In a previous video, the cells involved in the adaptive immune system were introduced. In this video, we will cover the mechanism by which T cells monitor the body. There are essentially two domains which the T cells preside over. The first are proteins made by self cells, and the second are proteins present in the extracellular matrix. Usually these are from pathogens. Each of these domains has their own respective receptor. The receptors that interact with T cells are called MHC receptors. MHC Class 1 is responsible for monitoring proteins made by native cells, and MHC Class 2 is responsible for monitoring proteins present in the extracellular space. All cells in the body, except red blood cells and several select tissues like the syncytial trophoblasts, have MHC Class 1 because it is used to monitor for viruses, cancer, and foreign material, processes that can affect any cell in the body. This MHC Class 1 pathway begins in the cytoplasm with the proteasome. The proteasome is involved with protein turnover and non-specifically degrades proteins into units of approximately 8 to 10 amino acids in length. These peptide fragments are then transported into the rough endoplasmic reticulum using a transporter TAP and loaded onto MHC Class 1. MHC Class 1 is exported via the Golgi system to the cell surface, where CD8 T cell receptors interact with the MHC Class 1 peptide complex. A key concept is that MHC molecules bind peptides in an extremely specific manner. They can detect the difference between just one or two amino acids at critical positions in the peptide fragment. This means that a given MHC receptor can only bind to a limited number of specific peptide fragments. Understanding this selectivity and process of MHC presentation is essential for understanding autoimmunity and pathogen evasion. We will cover these concepts in future videos. For now, knowing that almost all proteins made by a cell are broken down, their fragments are attached to MHC Class 1 on the rough endoplasmic reticulum, and they are presented on the cell surface for CD8 cell monitoring is sufficient. The second domain, monitoring of proteins in the extracellular space, is mediated by MHC Class 2 receptors. This pathway differs from the MHC Class 1 pathway in that protein fragments are derived from proteins degraded in the endosome-lysosome pathway. Because of this, the MHC Class 2 receptors are located on cells that regularly phagocytose pathogens. These are called professional antigen presenting cells, or APCs for short. These cells continuously endocytose the environment and present available protein fragments on MHC Class 2 for screening by CD4 T cells. From start to finish, the antigen presenting cells endocytose products of the environment. These endosomes are then combined with lysosomes. The proteins are degraded by lysosomal enzymes and loaded onto MHC Class 2 molecules. An important point in this pathway is the invariant chain. The invariant chain is a placeholder within MHC Class 2 that prevents proteins from binding during the folding and transport stage. In an endosome, the invariant chain is removed and protein fragments are bound to the MHC Class 2. Due to the location and the binding pathway of MHC Class 2, all proteins on the MHC Class 2 originate from the external environment. MHC Class 2, in combination with the corresponding peptide fragment displayed on the outside of antigen-presenting cells, interact with CD4 T cells. To conclude, we will quickly review the essential information. All proteins made by a cell are degraded by the proteasome non-specifically. Protein fragments are transported to the rough endoplasmic reticulum. If there is an MHC Class 1 that can bind specifically to a protein fragment, it will be loaded and then exported to the cell surface for scanning by CD8 T cells. Proteins found in the extracellular space are taken up by antigen-presenting cells and presented on the MHC Class 2 for scanning by CD4 T cells.